Kia ora and welcome to COVID Chat with me, Dr. Siouxsie Wiles. If you're joining us for the first time, I am a microbiologist and I'm here to talk a little bit about the science behind COVID-19 and to answer some of your questions. And speaking of questions, the person wrangling those is Damien Christie. Say hi, Damien.
Hello, how are you? I hear is there a cat somewhere in the set today?
She's gone again, but I'm sure she'll be back any moment.
Oh, I thought we had an emergency cuteness alert that we could. Well, if she does come back in, then by all means bring her onto the show. I know everyone's enjoying seeing animals at the moment to help them get a little bit of the warm fuzzies. So if you do have questions during the show, you can either, depending on where you're watching, you can either add them on the Facebook link if you're watching the Aotearoa Science Agency Facebook link, or otherwise send them to nzcovid@gmail.com. We're getting about 100 questions a day at the moment. We're trying to get through as many as we can. We're trying to group them into sort of broad topics and look at some of the things that we're going to be talking about on future shows, but we do try and get through as many of them as we can. But you'll appreciate if we don't get yours today, but it doesn't mean you shouldn't ask them. Siouxsie, should we start by having a look at what's been going on today?
Yeah, so lockdown, I think it is. And we woke this morning to the very sad news that one of the most privileged and powerful and wealthy men in the world, who has no underlying health conditions, is in intensive care with COVID-19. So if you think this is just a cold or the flu, think again. And we wish you a very speedy recovery, Prime Minister Boris Johnson. So yes, getting back to the cases, today there were 54 new confirmed and probable cases of COVID-19 here in New Zealand. What we really like to see is this plateau. So we aren't seeing exponential growth in the number of cases here in New Zealand. And that is because we have been in lockdown for 2 weeks, but also had some sort of stringent measures in place before this lockdown. What we hope to see now is these numbers of cases dropping every day. But these do bring us to a total of 1,160 cases here. So still a lot more than we'd like, but a lot less than lots of other countries. Speaking of other countries, globally, cases are growing at an alarming rate. If we look at the Hopkins tracker, over 1.3 million confirmed cases worldwide. The USA has overtaken Italy and Spain with almost 370,000 cases and shows no signs of slowing down yet. Over 74,000 deaths. This is clearly not a cold. Anyway, so today I want to talk about the race for a cure. You might have actually already heard that the race is already over. That's according to President Donald Trump, who's been talking about two drugs a lot, chloroquine and hydroxychloroquine. These are well-known drugs used to treat people with malaria, but also with the autoimmune conditions lupus and rheumatoid arthritis. And so I guess the question is, are these a cure? Do they really work? So the reason that he is saying they do is based on a pretty badly designed trial on a small number of people who had very mild symptoms with COVID-19, which was basically published actually in a journal. And the journal has since said, this perhaps didn't meet our standards for publication. But anyway, they haven't retracted it yet. But these people had mild symptoms, and I think from the paper it said that their symptoms got better about 24 hours earlier than those who didn't have the drug, which is pretty poor evidence for this actually being useful. Another small trial, but much better designed this time, has also just been published. That was done on people with much more severe symptoms, and they found there was absolutely no effect of having these drugs. The important thing, though, about these trials is that these drugs actually can have serious side effects, including kidney and liver failure. So it shouldn't be taken lightly, using these drugs. In fact, another paper that's sort of just been sped through in terms of being published was a study done on mice and showed that if mice were given this hydroxychloroquine, as well as a diabetes drug called metformin, that 30 to 40% of those mice died. Now, these people are actually studying the use of this drug to treat pancreatic cancer and using mice as their model animal. And so this was a really unexpected finding. But given that people have started talking about this drug as being a treatment for COVID-19, they rushed through publication of their results because they wanted people to know that this might, using this might be quite dangerous for patients. Terms of those drugs, which means that the race for a cure is still on, which brings me to telling you about this incredible, clinician, he's an epidemiologist and scientist called Dr. Ben Goldacre. You might have heard of him. He wrote a couple of books, Bad Science and Bad Pharma. He runs the evidence-based medicine lab at the University of Oxford, and he is part of a centre which is all about evidence-based medicine. And so what he has done, here's the centre, and they've been doing some really good stuff, analysing what evidence is coming out and then putting these little summaries up on their website. But what Ben has also done, so Ben is really interested in making sure that trials are properly recorded, and then that you can see the results of whether those trials have actually been published. And this comes from his interest from the fact that lots of pharmaceutical companies have hidden data from their trials of drugs if they didn't really go the way that they wanted them to go. So he's put together this COVID-19 trial tracker, which is basically tracking every clinical trial that's been registered for COVID-19. And there are 160 trials currently registered on the tracker as involving some kind of drug treatment. There are others, there's about 160 that also involve traditional medicine, but there are 160 that are involving different drugs, including 9 that are to do with hydroxychloroquine. But there are lots of others. So there are drugs, there's a couple of antiviral agents currently being trialed, remdesivir and favipiravir, which are drugs that stop the influenza virus from replicating inside infected cells, but also the use of antibodies from recovered people, as well as oxygen, and even a vitamin C trial. So these are all on the tracker. You can see what details have been given about what trials are and how many. I say is that some of them are very poorly designed, and it's unclear whether they'll actually give any good evidence for whether their treatment works or not. But there will be some really good, well-designed trials, which is exactly what we need at the moment, because it's through well-designed, controlled trials that we'll actually find out what suitable treatments there are. And this is tricky because obviously lots of health systems where patients being treated are under immense pressure at the moment because the number of cases has been rising exponentially. So to be trying to do a clinical trial, a good clinical trial amongst all that will be hard, but it can be done. And there's also been sort of questions about, how do you do a trial when you're trying to save people's lives? And so I guess I want to just very briefly touch on the fact that during the Ebola outbreak, most recent Ebola outbreak, they did do a trial of 4 different drugs and stopped it early because 2 of the drugs looked really promising. And so it would be unethical to withhold that treatment from other people. So there are ways of, evaluating the study as you go along. And then if some of the drugs look really promising, moving people from less promising drugs to the more promising drugs. So I think we're just going to have to wait and see. And what we want is really well-designed trials that actually answer this question, rather than just, trials, badly designed trials with drugs that are actually dangerous. So the hunt is still on. Right, that's drugs. Let's go and answer some of your questions, shall we?
Yeah, okay. So just a reminder, if you do have questions, you can send them to nzcovid@gmail.com. We'll get through as many of them as we can. We try and stick to the questions that have a scientific answer or some sort of based in science. So any questions about, do we think such and such is doing the right thing, or should we be doing what another country is doing? Although I would point out, Siouxsie, that you were I saw you on the social media talk today talking about the Swedish example. Can we just talk briefly about what's been going on there? What was the difference between Sweden and New Zealand, and what's happening now?
Well, so Sweden was going down the herd immunity route, which was, suggest some things around washing hands and social distancing and things like that, but don't do lockdowns, and just slow things down. And it's many people have been using it as so Sweden managed to keep their cases low. This must be working for them. What I said I had less than a week ago when this was raised by somebody as evidence for why New Zealand shouldn't be in lockdown. I said, why don't we wait a little while and see what's happening? And their cases are now rising exponentially, and they have more deaths than neighboring countries. And so I don't think they should be held up as an example for how to deal with this virus. But it's a very interesting, almost psychological thing that why do some countries think that things will play out differently there when, compared to their neighbors? It's kind of fascinating. We should probably get a psychologist on to answer that question for us.
We probably should. I think also one thing I've seen from people is that, this is potentially going to be afterwards a really good in the most terrible way example of a comparative study on different ways that different countries deal with the same problem and the outcomes. There's going to be a lot of evidence around, isn't there, after everything sort of settles down?
Yeah, but it's also going to be really complicated, right? Because there's lots of people saying, why have we done exactly the same thing as some other countries have done? And that'll be because there are differences in our governments. There's differences in, the level of compliance of people, of how, of how heavy-handed some governments are, how accepting people are of actions of government. So, it is going to be complicated, but it's going to be, yes, there's going to be years and years of study of teasing apart all these things. Who did what? When did they do it? Why did things work and why did things not work? Which is research, I guess.
Okay, we're going to jump to some questions. Question from Sonia. How can I tell which disinfectants are antiviral? Most of them don't say it on the bottle.
Yeah, that's a tough one. So there are a couple of things that you can look for. One of those would be whether it's got hypochlorite in it. So that's one of the main ingredients of bleach. So check whether hypochlorite is there. And usually when something says it's a disinfectant, it will probably have some activity. And then the other one to look for is activated hydrogen peroxide. So again, with both of these things, there should be all of the major brands of cleaners should have something in that range. If it just says it lifts fats and stuff, well, that might have some effect, but I would look for something that says disinfectant.
I've got a question just fresh off the press from Jessica, who's watching on YouTube. Do you feel that the small proven impact of vitamin C on cold duration and symptoms is encouraging as a possible treatment for COVID-19?
Oh,
Sorry, I've dropped you in
I
on
would
that
want to
one.
look at that one. I think that is one where there's lots of conflicting evidence on it from badly designed trials. So I would not like to comment.
Okay, we'll get back to you on that one, perhaps, Jessica. That's what happens when we
Yeah.
take questions live. You see, we don't know which ones you know the answers to or not. Question from Lauren, and now this is following on from a lot of discussion around masks, the new thing that people seem to be asking about. Are single-use plastic gloves from the supermarket good to use when you're outside doing groceries or pharmacy trips or the like?
I would say no, because, well, you don't need them. The glove is just acting like another layer of skin, right? So it's just to stop things from getting on your skin. But frankly, it's okay to get the virus on your skin, providing that you wash your hands. And what often happens with gloves is that people will feel a false sense of security. And as they take them off, they might then actually contaminate their hands and then not wash their hands afterwards. So I guess if you're wearing gloves, you might be more aware of when you touch your face. But I think on balance, it's still better just to wash your hands. And I've also heard of people leaving, like wearing gloves and then dumping them in the car park of the supermarket. So you're now leaving somebody else to pick them up and putting them at risk. So just wash your hands when you get home.
Okay. Question from Matt. New Zealand has an unusual level of exposure to UV. Is this an advantage against the virus in terms of fomites on, is that how you pronounce that, on outdoor surfaces and aerosolized virus particles hanging around the air?
Do we have a higher UV? I'm not actually sure about that. I mean,
What
I
can
would
I
have
answer that
said
one? I might.
maybe. I mean, does Australia have as well? Sure, answer it.
This is something that I've actually had a look in myself. I'm just a warning. I'm not a scientist. I do not have any science degrees, but I've done some googling on this before, and it's actually a bit of a myth that New Zealand has higher UV. The whole ozone layer hole being over New Zealand is not the case. It's actually much more over the Antarctic, and the reason that we tend to have higher rates of certain things, we do have cleaner air, and we spend more time outdoors, but generally speaking, do you know whether UV itself has an impact on the virus?
I think there are some people looking at this as a potential disinfectant, but I don't know enough about that. So, yeah, not sure about that.
I love this one. This is from Rebecca. I'm wondering if there's a good way to sterilize my paper copy of the New Zealand Herald each day. I know I should go digital, but I'm reluctant to give up this part of normality. Waiting 3 days wouldn't be fun because then it would be an olds paper. I'm currently microwaving it for 30 seconds. Will that work? I love that, Rebecca.
I I don't know. I would say wash your hands. Read your paper and then wash your hands afterwards. That's much better than putting it in the microwave. I know you might start a fire. Would you start a fire in a microwave? Not sure.
You could blow up your you can blow up your microwave. I've seen that being done before. We've got a question from Sharon. Does the amount of
Don't blow up
virus
your microwave,
particles
please.
Does the amount of
Sorry,
virus
Jamie.
particles you get on your body or exposed to determine how sick you get, i.e., one tiny particle compared to hundreds? This is about viral load, isn't it?
Yeah. Actually, so this is something that's not really known at the moment. You could certainly imagine that if you got one virus particle inside of you, then it would have, it would infect one cell, and then that would amplify the amount of virus. But if you had it in 20 cells, then that would have, they'd all then be doing their job and producing more virus particles. So it's not clear at the moment whether actually this is whether this is really important. It might be. It's been said as maybe one reason why some people are having a mild disease and others not, but it could just as equally be to do with our immune response or a whole bunch of other things. Those are the kinds of studies that are often done on using animals. So when you can actually completely control the dose, you can answer those questions. And so that may well be something that now the virus has been isolated, that scientists are going to start to work on. So I do know that some labs have started looking at, I think, ferrets as a model host for this virus. It's the same animal that's used to study influenza and a few other viruses like this. So that may well be something that they start doing, and that might give us some interesting answers to that question.
Here's a question from Paul. Is the aim of eliminating COVID-19 in the New Zealand context really achievable? And what does elimination actually mean?
That's a good question. I mean, it is achievable. If we can get everybody who has the virus isolated and allow them to go through their, the process of mounting immune response and clearing the virus, then in theory, there should be no virus left in New Zealand. But at some stage, we're going to have to open our borders. And then the question is going to become, what happens then? Do we have a 14-day, period where everybody coming in has to be quarantined? Do people who coming in have to have some kind of certificate to say whether they're virus free? That's going to be the problem, is reintroduction, I think, into the country. And maybe also how quickly people want to get out of lockdown, how much people are disobeying the rules. We've certainly heard a lot of high profile people disobeying the lockdown rules recently. All of these things impact on our ability to eliminate and then keep the virus out or at bay.
So we've got a bunch of questions coming in on the Facebook, and we will get to some of those in future shows. But that's about all we've got time for today. If you do have questions, remember, send them on to nzcovid@gmail.com. But yeah, our time is up today. Thank you so much, Siouxsie, for coming and helping us out with this again. I think maybe in the next show we might talk about there's been questions about vaccines. There's been questions about how do you keep safe when you go to the supermarket. We're definitely going to look at some of these things in upcoming shows, but we'll be back tomorrow at around about the same time. In the meantime, I thought we'd just go. I want to say thanks to the Science Media Centre for making the show possible. And I thought we'd go out looking at a rather ingenious way that police overseas have keeping people off the streets. Let's have a look at this clip just as we go out.
One.
Thank you.